Hello, everyone. I will speak about protein language model. So the first thing is to say a few things about proteins for people, especially from the laboratory that engineer that might not be familiar as I was. So first of all, proteins are large molecules that comprise long chains of amino acids that are basically organic compounds that contains amino and carboxylic acid groups. So the idea is that we have this chain, this sequence of amino acids, and fortunately for us, although there are over 500 amino acids in nature, only 20 are the ones that are relevant for us for biophysical applications because they are incorporated into proteins. This is quite good because we have a kind of an alphabet, so we have 20 tokens, 20 letters, from which we can build a language model and use the same tool as we would do for natural language processing. There is a difference between letters and words and sentences that are relevant for, let's say, ChatGPT and proteins. And the reason is that proteins have a structure. They can fold, and so only once the protein has folded, it becomes biologically active, and it has a specific biological function. So here with this slide, I would like to say one single concept is that proteins are a sequence of letters of amino acids. They have a structure and they have a function in the organism. These are the three main concepts, and I will try to get deeper in the following. But since I'm speaking about structural alphabet, I have to say a few more words about structure. And there are typically 4 classifications for structures of proteins, and the primary one is just the linear amino acid sequence that basically determines the native conformation of the protein. So it's just the sequence of amino acids. Then we can identify a secondary structure that is characterized by alpha helices and beta sheets, and they fall due to intermolecular hydrogen bonds, and these hydrogen bonds help stabilize are very helpful for the protein stability. So I have the amino acid sequence, and the first structure that I see due to hydrogen bonds are the beta sheets and alpha helices, and this is the typical picture that you will see for proteins. Then there is another contribution. So we have a tertiary and quaternary structures that are due to the fact that alpha helices and beta sheets have hydrophilic and hydrophobic portions. And for this, basically what happens is that the hydrophilic sides are typically facing the aqueous environment that is surrounding the protein, whereas the hydrophobic sides are facing the hydrophobic core of the protein. So for alpha helices and beta sheets then fall into a ternary structure due to these hydrophilic and hydrophobic bonds. They can assemble some units, and the tertiary structure may also give rise to a quaternary structure due to this folding of these subunits that are here. There is an example of the hemoglobin. But the relevant structure is for us the first, the second, and tertiary structure that are the one that will be modeled by this structural alphabet. Then I would like to introduce a couple of more concepts, and the first and most relevant one is the homology. So the idea is that we have sequences, we have structure, and we have functions of proteins in the organism. And we would like to identify what is the functions of proteins. And to do that, the main concept is homology. That is what we want to do is to identify evolutionary relationship within the protein domains. What happened is that there was a specific domain, a protein domain. It has some function in the organism. It starts, due to evolution, it started to mutate. But since it has a specific function, then even though the protein has mutated, the function and the structure are conserved. So there are clusters or families of proteins that, even though they have different sequences, they have the same structure and the same function inside the same biological function. And we would like to identify which proteins belong to which families and what is the main structure of these families. There are endlessly curated family databases and resources like PFAM, but the problem here is that we know millions of proteins and we cannot think of having manually created databases for such a huge protein databases. And for this reason, unsupervised machine learning tools can be very useful. I mean, we are not the first that are developing unsupervised machine learning tools for protein clustering and classification. And typical standard point is to cluster proteins starting from the sequence similarity, which is quite good because typically for proteins that have a sequence similarity at a level of higher than 40%, this is quite true. That is, proteins that have more than 40% sequence identity have similar structure and have similar functions, and they belong to the same families. The problem here is that it's not trivial to classify proteins that are in the twilight zone, meaning 20–30% of sequence identity. For this, what can happen is that due to evolution, amino acids start changing, and but the structure is similar. Upon evolution, so different sequences that are in the twilight zone cannot be identified with standard tools based on sequence similarity within the same family. But in fact, they have the similar structure. So the idea here is that for proteins within the twilight zone, a sequence alignment can be enough to detect proteins that have in the same family. And we would like to cluster not based on sequence, but directly based on structures. So the point here is that we would like to move from a sequence aligners to a structural aligners to identify to classify protein within the twilight zone. And there are such these tools are available, but unfortunately, clustering by structure is a non-trivial task. It's very computation expensive, especially for very large databases. So the idea is we need structural aligners, but we need them to be fast enough to be able to classify structures of millions of proteins. Trying to be inspired by the sequence aligners. So the idea is to try to have an alphabet that encodes the protein structure and use an aligners, a sequence aligners, but not on the letters that are the amino acids, but on some letters that encode some structural information. This is the main goal, to be fast enough and to use similar tools as what we typically do with sequences, and I mean amino acid sequences, but encoding some structures that allows us to classify protein within the twilight zone. This is the main goal. There is another thing that I would like to mention is that, again, we have protein language models that are basically they are the same thing as what. So first of all, let's started with the language model. So with words, we have BERT, we have GPT, and then they start saying, look, we have tokens. You can use the same tools as we do for language models, but apply for amino acids and protein sequences. And for this reason, there were lots of models, and one of the most relevant one is ESM by Meta people. That basically by masking, the idea here is to start to create a representation of the amino acids, and such a representation encode relation between amino acids, whereas ChatGPT creates a representation that encodes the relation between words. And in this way, we can learn something from the amino acids, such as prediction of mutational effect and long-range concat prediction. So here is how ESM works. So basically, we have a sequence of proteins. We start masking tokens, and we ask the neural network to reconstruct what is the token that I'm masking. And within this reconstruction procedure, the neural network starts learning relation between the amino acids. And within the self-attention mechanism, it is also able to learn the concat maps. So this is one of the largest achievements in protein language models. And as I was mentioning, it is not only giving us the opportunity for reconstructing the concat map, but it also can give us information about the protein stability upon mutation. So what can happen is that we might have a single point mutation that prevents the protein to fall into its normal native state. And we need a measurement to say when a protein is stable or not. And this measurement can be traced back to the change in the Gibbs free energy. The idea is that we have a folded state, an unfolded state, and a folded state. And there is a conformational change in the Gibbs free energy between these two states. And so we can compare the change in the free energy between the folded and unfolded states, between the wild type and in the mutated protein. And this difference in the free energy is an indicator of whether the mutation will be favorable or not favorable. So basically, here we have the folded state, the unfolded state, and the folded state. There is for the wild type in black, and there is a difference in the free energy. The same happens for the mutated, so the folded and unfolded state for the mutated protein. And then what we do is to compute this difference between the two free energy. If it is a negative, the difference in energy is less than 0, so we have a favorable mutation. Instead, if it's positive, then the mutation is not favorable. So the idea here is to use a pre-trained language model. So the model starts learning the representation of the protein, and then once we have the representation of the protein, we ask for the neural network to perform a downstream task and start to predict the delta G. So here the idea is that we have first a pre-trained unsupervised language model, and then what we do is to perform a supervised downstream task when we show two proteins and we give them an example that is a delta G, and the model starts learning, and then we ask a new protein, what do you think? And then the model will reply whether the protein is the mutation is stable, if it's favorable or not favorable. This is the main idea. And the idea here, this model, okay, it's a funny name. It's called Prostata. It's quite recent. It's something that appeared. It was published in November, and it reaches state-of-the-art performance computing this delta G. So now I want to answer to three questions. First of all, okay. So I would like to answer three questions. So what are these structural alphabet? How they can be useful for homology? And how can they be useful for thermodynamic protein stability? Okay. So the idea here has been introduced by the people from Foldseek. And the idea basically is to use an alphabet of 20 letters, 20 tokens, that somehow discretize the structural interaction between amino acids within the protein. So we would like to basically have an alphabet that describes the tertiary contact between nearest neighborhood within the 3D structure. The problem is that it is easy to have a notion of nearest neighborhood within a 1D sequence because it's just the next token. But since the protein fold, we need to understand what are the nearest neighborhood within a 3D structure. So for this, for each residue I, we need to select the one that has the smallest distance, but between virtual centers. So first we need to optimize, find these virtual centers and find the one that are the nearest neighborhood within this 3D structure. And the model is optimized in such a way that it typically prefers to select long-range tertiary interaction compared to the backbone conformation. So basically, the model identifies the nearest neighborhood as preferring the one that are farther away in order to enlighten the 3D conformational structure rather than choosing just the next one, because otherwise it would be too simple and would not capture the 3D structure. So there is a optimization procedure to select which are the nearest neighborhood in a 3D structure. And once we understand what are the carbon alpha that of interest, so I and J, we define 5 vectors. Basically, once we find C, I, and J, we define 5 vectors, and we start defining the distance between I and J and other 8 angular variables that define the local 3D interaction. Once we found these 8 angles, 7 angles, and the distance and other couple of variables that describe the local shape of the interaction, the idea is to use a quantized variational autoencoder to map these 10 continuous variables into 20 letters. And how does it work? It works like here. So we have our 7 angles, the distance, and other couple of variables. We perform an encoding and we project our 10 variables into two-dimensional plane, Latent space, where we cluster the representation, find the nearest centroid, and each centroid represents a letter in the Latent space. And then we ask from this Latent space to get back to the physical description. So the idea is that we want to translate angles and distance between amino acids into basically compress the information into 20 discretized tokens, 20 letters, and then reconstruct back the physical descriptors. In this way, we are able to compress the information. So we have a 3D structure. We want to compress the information, squeeze it into 20 discrete letters, and then get back and reconstruct the original structure, the original angles. By doing this, we are learning to encode a continuous information into a discretized set of tokens and get back. So we have a mapping, a translation between say, structural continuous variables into a set of tokens that basically will play the role of language modeling. So now we have, within this way, we have a sequence of letters, but these letters do not represent amino acids, but represents discretized angles, say, or discretized distance between residues. What we can do is we start clustering. So we have a sequence of letters at this point. We can define a distance between this sequence of letters, and the distance basically is the overlap. If the two regions of sequence overlap, they are nearby. If they do not overlap, they are far away. And once we have a notion of distance, we can also have a notion of density, because let's say in a radius of 1 meter, if I have 5 people, I have such one given density. If I have only 1 people in the nearby, I have less density. So having a notion of points within a distance, I have also a notion of a density. And once I have a notion of density, I can start clustering based on that density. So here is a pipeline that is based on density peak that has been developed by Russo, Barone, Laio, and Marco Punta between SIS and ARIA in the last few years. This is the algorithm. More pictorially, what we have is something like that. So we have a protein. We have a distance between protein that is based on the overlap. And upon the overlap, we have a higher density region and less density region. And higher density region will be clustered. And the cluster of these structures will be the families that will have a similar structure and similar function within the organism. So here is the idea. So we would like to find proteins that have a similar structure and similar function. And to do that, we start clustering and using these unsupervised machine learning tools that will help researchers to have a clue of where to look at. And it is called DPGstruct because at the end of the day is a homology, because it will search for homology. Density peak clustering, it is an algorithm that performs clustering based on density of structural local alignments. This is the main goal. We have some results so far. So on the left, we have seen that we have several indicators of whether our cluster really reflects homology. So we have the probability of homology according to scope. We have the LDDT and TM score, and we see that all these measurements are related. And also we see that smaller clusters tend to perform better, have a higher probability of homology Instead, the bigger cluster are we suspect that we are clustering too many different structures. So all these violet points are still under investigation. Instead, the yellow points are quite good. We are satisfied about this. We also see that we have a power law distribution on the upper right lane. This power law is quite similar to the zip law for language, natural language. So we have many clusters that are small and few clusters that are very large. Precise, like in standard words, we have many words that are shortened and very few words that are with, I don't know, 15 letters. And they follow a power law distribution, and we see that there is a typical agreement between clustering on sequence, clustering on structure, and across different databases, smaller database and bigger database. They all follow a similar power law. And we also see in the right lower plane that we have good agreement with manual curated databases like PFAM. So we have also consistency with PFAM that is higher than 95%. But those are numbers. Let's see. Let's pick here. What I did is just to pick 3 proteins within a cluster randomly. On the right, there are 3 proteins that are within the same cluster of A, and we see 3 beta sheet completely aligned. On the left, we see that we have a very big green protein, but if we focus on the domain, we see that is perfectly in line with the alpha helices to other 3 proteins. So there is a region in the green protein that is perfectly aligned with the blue and the red one. Here I'm just picking a cluster and I'm inspecting the proteins within the cluster and see whether they are really aligned and really belong to the same family. This was just an example. To conclude, I would like to address the other problem, is that how can we use this structural token, structural alphabet for protein language model? And here the idea is to, this is not our work, has been done in the last June, so it's something that also is from this year. By Eitzinger and by Steinegger and the group. And the idea here is to have 2 different alphabets. So say that we have a language model and we have, I don't know, English and Chinese. My language model shall be able to pass from one language to the other. I mean, language models have been first designed to perform tasks like translations. And here the idea is very similar, is to be able to perform a translation and to learn a language, understand the notion, the concept, and be able to translate that concept within a different language. So the idea here is to have a pre-trained language model that is able to pass from the amino acid sequence, learn the relation between the amino acids, translate them into this other alphabet that I recall is basically a discretized encoding of the tertiary structure. And here the idea is that once we have learned this, we can use to extract for feature extraction and to perform a downstream task, precisely like the prediction of the delta G. So here we wanted to use an augmented alphabet, incorporating also the 3DI structure to predict the delta G. Unfortunately, we have some preliminary result on the fine-tuning using POST5, this model, but we see a lower accuracy compared to other models like the MSA transformer. And the reason here is that an MSA transformer, when we create a multiple sequence alignments, we show the models many proteins that are not identical, many sequences that are not identical, but are they similar. So we think that the MSA transformer here performs better because we showed to the model what evolution basically produced. So showing the model an MSA, we are showing them many proteins that are stable, that are being evolutionary stable, and then when we showed it something else, it recognizes that it is something different. So for this reason, POST5 doesn't seems not to work that well compared to other tools like MSA Transformer. Maybe we can try different models. For instance, this structural aware protein SAPROT, SAPROT. It is another protein language model trained on this 3DI that appeared last October. We might try to use this, or we keep on going and perform again research. So my conclusion was mainly to introduce this structural alphabet because we think it might be a very interesting tool to exploit. It has, especially for the homology search, we have results and they are very promising. For protein stability, we are interested. We are continuing to do research, but for this, we also rely to other models like the MSA Transformer or ESM2. We are open and we would like to collaborate if possible. And I mean, we are here to use all the possible tools and be at your disposal for if you have experimental results or you want to share anything with us.